Great to have all of you here today. We're going to talk today about infection to pandemic, how viral adaptation between different hosts, the origins of viral outbreaks, virus transmission, cell entry, and tissue tropism. So we'll talk about these different phenotypical properties of viruses and how they might be detected at the level of the genome, the viral genome. And before we jump into that, I wanted to kind of review some of the things that we did up until now. I know that for myself, there are a number of different programs that I'm in, and it's difficult to remember exactly what's happening where. So I do want to start off with a review and also just kind of look at where we are as a group. So I have this as a reference. But in reality, I wanted to look at, how is everyone doing? I think that it's very important for us to keep track of where we are as a group. This is the program page. On this program page, you can see where, some of your activity, right? So the different coursework that you're completing, and if there's anything posted on the forum. So on Friday, I don't know if you got the invitation. I hope that you did. We do have this recording of the webinar. In the webinar, Dr. Koussoulis from the LSU School of Veterinary Medicine talked about the coronaviruses and specifically different approaches to vaccine design and different approaches to antiviral drug design. I think it was a fascinating lecture. I encourage everybody to take a look at this lecture. In the second part of the lecture, the overall recording is very long. It's about 4 hours long, so I'm not expecting everybody to watch it, but there is a second part there that talks about bioinformatics and the different approaches related to analysis of the novel coronavirus genomes and transcriptome that is available from the public domain. So if you're interested in those topics, please take a look. That is going to be on the forum there, right here. And in terms of progress, I want to, briefly to remind everyone that the objective of the program is to provide you with the tools and with guidance on how to start a project of your own and analyze data that we are reviewing. And we're going to be reviewing data from a multiple of different types of viruses. All of this data is publicly available, and hopefully by soon, in the next couple of weeks, you all will be able to apply some of the methods that we're learning about to a project of your own, really understanding how analysis of genomic data, of transcriptomic data in the host response could help us understand viral infection and bacterial infection, and also help us analyze different aspects of that infection and compare different outbreaks. So please review some of the materials that we've done up until now. Those include analysis of host transcriptome, where you can detect viral genomes. It also includes multiple sequence alignment of genomic sequences and analysis of viral diversity, how we can study haplotypes of different viruses. So in fact, I prepared a few slides to help review some of the pipeline outputs that we've done up until now. We've run a number of different pipelines, starting from visualization of read alignment to the reference genome, multiple sequence alignment, phylogenetic trees, and SHORA, which is a method to identify haplotypes and study consensus sequences of those haplotypes. So the first pipeline that I wanted to review is an analysis of Ebola samples, a couple of samples. One was viral amplicon sequencing, and another one was metagenomic sequences of viruses. And we aligned it to 2 different genomes. So I don't know if you remember, but we talked about how these are metagenomic samples from bat fecal samples, and one was specifically to amplify a specific viral genome, RAD-G13 genome. Another one would be to detect a presence of a viral genome. And so we could really use any kind of a reference genome. So to do that kind of a comparison, we ran two different pipelines. One pipeline, as you can see here on top, was mapping reads on the RAD-G13 genome. And in this example, you can see how the amplicon sequencing has a very small number of reads compared to larger number of reads for the metagenomic sequencing. But also you can see here individual positions that are different from the reference sequence. Remember that in the visualization, these little icons represent a consistent change from the reference. So in this case, you can see only one read is available, and if it contains a change, then that will happen with this label. And on the RAD-G13 genome, there's not that many mutations or not that many differences. Here, we can visualize also mapping on the novel coronavirus from the human. And you can see how many mutations start appearing right here, right? So this gives you a sense of how different these genomes are and how we can take, different mapping. We can use different references to really see what mapping gives us, what kinds of information does it give us. So we also can look at another output from this pipeline. In this pipeline, we have an output from Bowtie, and Bowtie gives us some statistics on the alignment quality, including how many reads align exactly to the reference genome and how many reads align 0 times, for example, or don't align at all to the reference genome. So whenever we compare, again, remember that these are two samples, right? So there's sample 1, there's sample 2, sample 1, sample 2. So we have the same two samples aligned to two different genomes. And here you can see that surprisingly, the RAD-G13 genome, which does not contain a lot of mutations, the overall alignment rate is actually smaller than if we align it to the novel coronavirus Wuhan SARS-2 genome. So here you can see almost 20% aligned to this Wuhan sequence, and here only 0.01% aligned. But if you look here in more detail, you can see that a lot of reads that align to this Wuhan SARS-2 genome actually align more than one time. And so the quality of alignment is not as good, right? And we kind of see that in the results of the visualization. You really see a lot of mutations here when you don't see a lot of those reads aligned. In the amplicon, there's not that many reads. There's only 33. And so all of those were actually aligned. 100% of those were impaired and 100% of those aligned to the reference genome. So I think this is an interesting comparison, but gives us really a visual sense of how data from next generation sequencing can be analyzed for reference according to the reference genome. Another type of a pipeline that we did is analysis, phylogenetic analysis, and analysis of these haplotypes. So again, hopefully some of you had a chance to reproduce this analysis on your own as well. If you have not, I suggest that you do it. I provided the files and they're also available on the forum. But here's a pipeline that I did from one of the previous times that we did this example. Here you can see that basically we start off with, again, these are FASTQ files. They're not assembled genomes. These are just reads from biological samples. And you can see how the result of this analysis of these haplotypes is actually assembly of variable genomes that are present in a sample. And the output is also a phylogenetic tree. The phylogenetic tree, as you can see, has a number of different haplotypes. So haplotypes 0, 1, 2, 3, and 4. So a total of 5 haplotypes in group 2, and a number of haplotypes in group 1. Let's see how many. 1, 2, 3, so I think 8, so 9 haplotypes in group 1. What are these groups? Well, if we look in the uploaded data, we'll see that group 2 was Sierra Leone and group 1 was Liberia. And so these samples, we know what they are by looking at, we know what they are by looking at the uploaded data. So the conclusion from this is that in Liberia, specifically this sample that we took from Liberia, the diversity of viruses, of the Ebola viruses in that sample was higher compared to what we saw in this sample in Sierra Leone. Now, this does not give us any kind of conclusion for overall what happened in those countries, because we are only using one sample per location, so really not a lot of power to this analysis. But again, it just illustrates how we can compare diversity of viral genomes inside each sample using next generation sequencing. And you can also see in the outputs that you also get consensus genomes, and those consensus genomes will be provided for each group independently, as well as for overall what might be a consensus genome from all of these together. So it's a useful type of analysis. If we have multiple samples that we organize per group, we can probably get a better sense of how diversity changes between different locations and how it changes between different types of samples. Another pipeline that we did was just a simple phylogenetic analysis. And this phylogenetic analysis was based on analysis of, again, samples taken from multiple different locations. Those different locations grouped together, right? So we can see here Liberia, UK, and Gabon. But another factor was time, right? And so you can see here the date was 2014 versus 1996. So really the Gabon sequences that were grouped together in this group 1 were earlier, much earlier, 1996 compared to the later. Now another kind of outcome of this analysis, right, is you see that this outgroup reference grouped together with these samples, it was closer to the 1996 samples or to the Gabon sequences rather than the 2014 sequences. What does that tell us about this reference genome and these samples? Well, to understand a little bit better what that means, we actually have to look at this reference genome to understand where did this data come from. So if we go to the sequence, we'll be able to see, this is the link, right? I'm going to put this link in chat for you all to see as well. If you look on this, oh, sorry, I made that private, so let me just give it to everyone in the session. So if you look at this link, please tell me what date was this sequence, what date was it from? So what is the year that the sequence was collected in, this genome was assembled in? Okay, 2018 is when it was published, that's correct. But when was it actually collected and assembled in? Okay, anyone else? Okay, so you can actually see right here, 1976, right, is the time when it was collected. So you can also go to this link, which is the Bio Project. The Bio Project actually tells you how this data was, produced. And you will see here that they did a number of, it's a review of previously collected data sets, and that's why you see the date of 2018. That's the date when it was registered. But in fact, it is based on genomic DNA from a whole number of different samples. And so if you look over here, this is the Ebola virus from the host, Homo sapiens. TC stands for tissue culture. COD stands for Côte d'Ivoire. And 1976 is the time when it was, the sample was collected. So this is an earlier reference genome, and that's in fact what we see here, is that this specific genome actually appears to be closer to this earlier set of samples from Gabon. And then we talked about how a similar type of an analysis, but in fact a number of additional methods, are also organized in this other section called virus quasi-species. And we'll talk more about the impact of diversity of viral sequences on the phenotype of disease, on disease severity and disease progression or virulence. And we'll talk a little bit more about different methods that help us identify these unique sample variability in the genomic sequences. So virus quasi-species is a section dedicated to assembling all of the relevant methods that help us study viral mutations. And so you'll see here analysis of point mutations, analysis of defective viral genomes like deletions of whole genes and regions in the viral genome. And you'll also see analysis of viral quasi-species and their phylogeny, how they are related to each other evolutionarily. So in that section, we did an analysis of coronaviruses, and these coronaviruses were also organized by different groups. And so we'll talk more about this pipeline in the context of how is the virus different between where it originated, as well as how it is different between other strains that were previously recorded infecting humans. And potentially the relationship between the origin of the virus, the intermediate host, where the virus might have recombined with other similar genomic strains of that virus, and how it is manifested in the human population. Maybe all of those things are related. So we'll talk about that. But we also talked about how some of the outputs from this kind of a pipeline could be visualized, right? And so it's not just the phylogenetic tree, the dendrogram that we were just looking at. It's also ability to look at individual positions on the viral genome. We can collect them by bins and see at frequency of mutations in specific bins or regions of the genome. We can map those frequencies and diversity onto the protein sequences. And we can also look at how they are related to each other. Are they more closely related to each other, or are they more distant between each other, etc. Underneath this kind of a high-level view is really level of sequence, right? And so here you can see different tools that could be used for study of sequence similarity. So this is just sequence alignment. We can look at sequence similarity. I gave you a number of different methods that you can use to visualize data in such a way, and the output would be FASTA or Clustal, and we looked at how those files could be visualized. And then there's also tools that help us understand the relationship between sequence similarity. Here you can see sequence similarity in amino acid sequence and relationship to the structure of a certain protein. So in some future lectures, we'll actually do a hands-on analysis of detection of similar regions and comparison of similar proteins that we can superimpose on top of each other to understand how related they are and how close they are in terms of their interaction with, for example, human receptors through which the virus enters into the host cell. So were there any questions about some of these pipelines that we've run up until now? And again, I know the question about the coronaviruses, and I'll get to that in a second, but yeah, so Roland has a question. Go ahead.
My question is basically like in these data sets, like and upon looking at the links of each read, is it possible to like is it possible to use like RNA sequencing upon looking at the length of each read, whether it's a short read or a long read?
Okay, good question. So the question is so there are two questions that I see in what you just mentioned. One of them is whether we can do analysis of RNA. So for example, mRNA expression, so analysis of transcriptomic data. And another question is maybe about read length. Is that correct?
Correct. Yes, sir.
Okay, so yes, definitely the same next generation sequencing technology applies to the study of RNA sequencing or the study of RNA or mRNA expression. The difference is between DNA and RNA sequencing is not in how the sequencing is done, it's more on how you prepare the sample. What is your source of data? Is it RNA? Is it mRNA? Or is it DNA? For example, for DNA sequencing, we might choose to extract the DNA of the, let's say, the human DNA, and extracting that DNA will then give us, if we sequence all of it, will give us whole genome sequencing, right? So we will understand the sequence of that DNA. In terms of RNA sequencing, we don't extract the DNA, we only extract the RNA. And sometimes we want to only extract the RNA that is being produced, that is being translated into mRNA. So that will be called poly-A RNA sequencing. So we're only sequencing those strains of RNA that are ready to be used by the ribosome and translated into protein. So we will, we can use the same sequencing technology, but really it's the material that is being put in that determines what are we measuring. The analysis will be different. So when we look at DNA, we typically look at obviously organisms that have a DNA. So coronaviruses, for example, would not apply. Can we detect the presence of viruses in RNA sequencing? Yes, because many viruses use the host cell machinery to produce their proteins. And so it's likely that we will find RNA from viruses and find that in host RNA if we do sequencing of mRNA, and especially if we do total RNA, so if we're looking at all of the RNA present in the sample. How does the read length apply to the study of mRNA or in fact, next generation sequencing in general? Read length really helps us understand where the sequence came from. So the longer the read, the more confident can we be about its location on the reference genome. For example, many reference genomes will have repeats of certain genes, or certain segments might look very similar. So the longer the read, the more confidence we can have about where they're actually coming from in reference to the genome of the host or whatever genome we're using. So that would be my answer. Is that clear?
Yes, sir.
All right, great. And then did anyone else have any other questions? Okay, well, if you have any other questions, put them in the chat. I'll be happy to answer those as well. Okay, so today's session, we wanted to talk about how does a virus go from just being a disease that's out there to becoming a pandemic. And so we'll talk a little bit about viral adaptation, and specifically we'll talk first about the Ebola epidemic, and then we'll also touch on coronavirus pandemic to talk about the different factors that differentiate viruses that are established in a host, viruses that transition between different hosts of different species potentially, and how these events can change the virulence of disease. And virulence is what typically is viral replication or associated with severity or harmfulness of a disease, as well as pathogenesis, so how the virus can cause disease and how the disease develops over time and symptoms that it produces. So as I mentioned, we'll start from Ebola viruses. For Ebola viruses, at a certain point, you can see here an article from 2016, and this is just one example. There was a lot of interest in identification of specific mutations that might have increased the virus's ability to go between humans, so transmissibility, and potentially even making it more deadly. And there were a variety of different speculations about mutations that can change, for example, the way the virus infects. Maybe instead of being passed from bodily fluids, it could become airborne, and that way it could be more dangerous because it would affect more people. In this article, it was mentioned that earlier studies of the Ebola outbreak in 2013 to 2016, there was a lot of emphasis on location and the specific location kind of unique aspects of the health system and density of populations, and many human behavioral factors that have helped the virus explode across the West Africa and eclipse any Ebola outbreaks that preceded it. And these new findings around the year 2016 actually complicated the story and suggested that the viral genomes and the variation in those genomes also have a part to play in the number of people that were affected by this virus and that died from this virus. And so they were looking at how the different mutations in specific regions of this virus and specific regions, geographical regions, could be related. So to understand a little bit better what they were trying to do, let's take a look again at next strain, and we look at the specific diverse populations of the virus and how they're associated with the location. So let's take a look. Okay, we'll go to, again, next strain, and I'll give you the link right here. So if you could also navigate to this link for you on your computer. And here, try to observe as we play this animation exactly how the virus spreads throughout these years from 2014 to the end of 2015. Okay, so you can see that it originated in Guinea. There are a lot of infections. Almost simultaneously, it spread in a number of different locations, separately in Liberia and Sierra Leone, right here. And you can see that there are multiple infections that went back and forth between the borders of these two different countries. Now, the colors are associated with the division, so they're associated with independent kind of clades on this phylogenetic tree. As you look, the tree is being built. Specific strains just simply died out, So for example, the orange strain and the light green strain, right, they did not exist past almost the beginning of 2015. And then some of these other ones lingered for much longer and even became more diverse towards the end. Right, and at the end, if you kind of take a look, there were only two that were left. This one right here and this one right here. Okay, so based on what you see, what would you say was the most infectious, or the strain that affected the most people? If you look at the different colors, maybe if you look right here on this side, what would you say, what color affected the most number of people?
I see a lot of purple, so I would go with purple.
Okay, great. Purple, so at least a couple of you think it's purple. I agree, so purple seems to have these larger circles right here. And what would you say about the one that is maybe the least, affected the least number of people?
Red.
Red, excellent. Okay, so here we do see just a couple of, or maybe just a few dots of red, and you can also see that right here, it is present, but not as much, right? So we definitely see a lot of purple, we definitely see a lot of blue, and then you can see some of the smaller ones. So now if you look at these different types of strains, right, the basic analysis that we could do is to try and compare them to understand how the different mutations in these colors, in these strains, could be associated with the outcome, with what we're actually observing, right? And so here, if you noticed as this animation was playing, right? So let's play it again. Right, we see here the green, we see the orange, here's the blue. Let's pay attention to when the purple, okay, so here's the purple, right? So the purple did not come until later on, right? And so if we try to separate these samples, it's probably difficult to separate them by country alone, because blue and purple together were in Sierra Leone, right? So if we just take Sierra Leone versus, let's say, Liberia or Guinea, we will probably not get a very good separation, but we definitely see how the purple is associated with a much later phase. And there's a lot of these infections happening right here at the later phase as it continues to spread. And it seems to linger in this place, but also continue all the way until the end. So based on this, we can try to compare not just based on geolocation, but actually based on the time of the sample collection. Now, this animation is associated with data. And if you scroll down, you can see how you can differentiate it by country, right? So which country is this? This is Sierra Leone. Let's take a look at that one. And you can see the numbers right here: Guinea 365, Liberia 187, Sierra Leone 941. So it's definitely a country that has a lot more, as you can see right here, right? But they are unique. So they are actually not these, right? So if you kind of take these two, the light green and the orange, and you compare it to some of the other ones, you'll probably see a lot of variation. You can also see here that some of these early infections in Sierra Leone were, or sorry, not in Sierra Leone, was also associated with some of these. So what really is we can observe here is that right now, all of the colored ones are Sierra Leone, right? So let's play this animation again. Sierra Leone infections happened in multiple times. So they happened at the very beginning, as we see right here, and they were primarily blue. And as it progressed, there was more and more of the emergence of these purple ones. And then the purples became much more than the blue. So as you see the timeline going, you can see that it's becoming more and more purple. What would you say does this mean, or how significant is this for us to be able to take specific samples that then we can use as comparisons with each other? What might be your conclusions from seeing this develop over time? So again, anyone, you're welcome to jump in here. What might be some of your ideas about what this might mean about the virus? Okay, so I'll just summarize for all of us. Potentially what it means is that the virus really changed over time. So obviously before jumping into humans, it was present in some other host, right? We talked about bats and we talked about some intermediate hosts, potentially primates, but then it jumped into the human host. And we know that whenever that jump initially happens, the virus mutates rapidly to adapt to the human population. Once it finds an effective way to infiltrate the human cells and to transmit between humans, it will continue to develop and it will cause a lot more disease. So it will be much more severe in the very beginning, but at some point the viral objective, sort of evolutionary or metaphorically, is to find a balance between being very deadly and effective at replication and actually be mild in the sense that it does not kill the host and continues to replicate. And so what you can see here is that a lot of these kind of, the virus died out essentially in a short period of time, in under a year. And then here the virus continued to be present in the human population, becoming more diverse and also becoming more specific, right? So all of these are purple and they're associated with a specific sort of genotype of this virus. So let's take a look at how this might explain the biology of this virus. So originally, when we are looking at these viral sequences, we're looking at two different processes. We're looking at mutations, which could be random, so they occur randomly because of the viral genetics. And the ultimate source of genetic variation is point mutations. And then the second one is recombination. Recombination is a pervasive process that generates diversity in most viruses. It joins variants that arise independently within the same molecule, creating new opportunities for viruses to overcome selective pressures and to adapt to new environments and hosts. Recombination occurs when at least two viral genomes co-infect the same host cell and exchange genetic segments. Different types of viral recombination are recognized based on the structure of the crossover site. Homologous recombination occurs in the same site in both parental strands, while non-homologous or illegitimate recombination occurs at different sites of the genetic fragments involved. So you can see that specifically in RNA viruses, there need to be two different viral strains within the same host for a recombination to happen. The exchange of genetic material between viruses is usually non-reciprocal, meaning the recipient of a genome portion does not act as a donor of the replaced portion in the original source. In this respect, the term recombination does not have the same meaning in viruses that it does in diploid sexually reproducing organism, where the exchange of genetic material between chromatids in the first meiosis division is reciprocal. Viral recombination could be more appropriately denoted as gene conversion, but the term has become so widely used that it is pointless to change it. Recombination is a widespread phenomenon in viruses and can have major impact on their evolution. Indeed, recombination has been associated with the expansion of viral host ranges, the emergence of new viruses, the alteration of transmission vector specificities, increase in virulence and pathogenesis, the modification of tissue tropisms, the evasion of host immunity, and the evolution of resistance to antivirals. So when we look at these viruses, we are observing how the viruses exchange this genetic material. And the first question that we want to address is where the virus came from. So there's no real clear understanding on whether emerging viruses become more or less virulent following a jump to a new host. Before a host jump, pathogens are likely to have been selected to optimize their virulence for onward transmission in the reservoir host. However, immediately following a host jump, the pathogen is likely to be initially poorly adapted to the new host, which may inhibit successful emergence, and the host is likely to be poorly adapted to the new pathogen. High host mortality is associated with a reduced likelihood of successful onward transmission in the case of human viruses. There are three possible scenarios for virulence evolution immediately following a host jump. So here you can see several examples. One is when the virulence increases. For example, when it goes from monkeys to humans and it becomes more infectious, more deadly to the host. Another example is when it does not change or the virulence actually decreases. So here was killing a lot of fish, and here it only killed a small portion of fish, right? So this type of viral recombination and ability to jump between hosts is also related to how severe this infection affects the new host that it is in. So when we talk about this pandemic, the viral pandemic, the Ebola pandemic, we know that it originated in a single location. It jumped over into a new host, which is human, and then it started spreading between humans. And when we see the spread between humans, we see two major types of spread, right? The original one was quick and very and deadly, but it ended fairly soon. And then eventually over time, it changed to a more persistent type of an infection that spread to many more people in also a fairly short period of time. And the viral genome actually changed. So when we look at the insights, when we look at all of these different viral genomes collected throughout the whole infection, we actually see that we have a fairly clear picture of the origin and spread of this Ebola virus. One of the most important early questions was whether the epidemic was the result of a single cross-species transmission event into humans, or whether there were repeated zoonotic events from a widespread animal Ebola virus reservoir. Due to the high genetic similarity of viral genomes sampled from the beginning of the epidemic, a single spillover infection seems to be the most likely. Phylogenetic analysis also make it clear that once the outbreak was established, later lineages of Ebola virus Mucona have descended from those circulating earlier in the epidemic. This is in contrast to some of the earlier Ebola virus outbreaks in which epidemiological and sequence-based investigations have provided evidence for multiple spillover infections. So again, two scenarios. One scenario is a single spillover event. The second scenario is when there's a continuous spillover. For example, if you think of the virus infecting not only humans, but potentially infecting animals that live in close proximity to humans, cows, pigs, sheep, dogs, cats, etc., we can imagine how continuous interaction with those animals results in a number of repeated outbreaks And those might be also complicated by the fact that viruses in some of these intermediate hosts could be recombining. So viruses that are more effective at spreading in human population, potentially causing mild symptoms, recombining with viruses that cause high rate of deadly outcomes and high virulence. So as the continuous study of these viral genomes continued, you can see here, the viral outbreak was separated into 3 different lineages. And those lineages, we saw how they actually affect specific countries, right? And you can see here the number of cases associated with each one of these different lineages. So as we saw, right, the most was right here in the blue. And then right here, we have these separate 3 locations that were very different. One of the hypotheses very early on was there will be a specific mutation, not just anywhere in the genome, but specifically on this surface glycoprotein. And that might change the way that the virus enters the human cell. So for example, when we look at the surface glycoprotein, one of the the proteins in general, we can see that specifically the surface glycoprotein is actually the one that is responsible for entry into the human cell. So we can take a look at this protein right here. Let's take a look at this protein and we'll read about it. Okay, so getting into cells, the Ebola glycoprotein has the task binding to receptors on a cell surface and getting the Ebola genome inside. It shares many of the features of other viral fusion proteins, such as influenza hemagglutinin and HIV envelope glycoprotein. It extends from the surface of the virus and is covered with carbohydrate chains that hide it from the immune system. It is also a highly dynamic protein that snaps into a different shape when it binds to a cell surface, dragging the virus and cell close enough to each other that the membranes fuse. And the structure shown here is PDB entry 3 CSV, so we can actually look at that viral protein. It includes the receptor binding portion of the glycoprotein and the machinery for membrane fusion. To allow cystillization, a small domain has been removed that includes much of the carbohydrate that normally covers the glycoprotein. So let's take a look at this glycoprotein. Here it is. So you can see here the sequence. The sequence is in amino acids. And here's the actual structure of this glycoprotein. So you can see that it has this kind of a shape. Of course, this is not the full shape. The full shape has an additional portion down below that goes into the viral membrane. So when we talk about these different viral lineages, right? So let's take a look here. Let's identify specifically where is that protein. So which protein actually encodes for this glycoprotein. So here you can see this viral protein. Let's see real quick. Okay. The viral protein, the glycoprotein is VP24. Not VP24, the glycoprotein, let's see. Okay. So this is the glycoprotein right here. You can see GP stands for glycoprotein, right? And this shows the diversity specifically in, so these two are actually the glycoprotein, and you can see how enriched this whole segment is with mutations, right? So if we actually look at all of the data, okay, let's look at all of the data, right? And I think we did this in one of the first sessions. We can see that many of the mutations that occur throughout this diversification of the viral lineages actually occur in the glycoprotein. So what is it in the glycoprotein that actually makes this virus potentially more deadly and more severe as it spreads? Okay, we looked at the diversity, and it's important. I will provide this article, by the way, that I think helps us understand how diversity, right? When we look at specific mutations emerging over time, we typically think that there's going to be one single mutation that characterizes the difference. And to obtain that single mutation, let's think of this mutation as this one right here. This mutation is actually present in many, many different viruses, and those different viral genomes contain that single change. But when we look at how many of those viruses are in a single host, in a single patient, we should also consider how the diversity of viruses actually contribute to its ability to produce virulence, to replicate, to spread between different tissues, and to infect multiple people. So in this article that I provided you with here in chat, let's take a look at that article. There are actually two different articles that I wanted to kind of review in this context. An article that talks about a specific mutation in the glycoprotein that changes one amino acid, and the general article about diversity, right? So let me pull up this article right here. This is a review article about how high throughput sequencing can help us understand diversity of viral genomes and study the diversity of viral genomes to understand how the evolutionary dynamics of viral genomes help it evade the immune response, help it evade vaccine, so complicate vaccine design, because vaccines are designed to help the immune system recognize the virus, and potentially be more dynamic and therefore be more able to overcome specific pressures that are in different tissues. So you can see here, specifically this segment here talks about recombination that we already talked about, and also talks about the heterogeneous mixture of viral strains that appears to confer numerous advantages to the virus population, including the ability to escape from the host's immune response, the development of resistance to vaccines and antiviral drugs, and has significant implications for viral pathogenesis, virulence, persistence, and disease progression, and likely contributes to tissue tropism. The robust adaptability featured by RNA viruses, which is related to their genetic heterogeneity, is thus of clinical relevance. In fact, many of the infectious diseases which have jeopardized and still are a threat of public health are caused by RNA viruses, including HIV, hepatitis C, HCV, influenza virus, Ebola virus, and Zika virus. So again, what we see here are two examples. An example of diversity, how the diversity of viral genomes present in a specific group of viruses causes more virulence and potentially could explain how many people it affects in a short period of time. And the second one is really the emergence of very specific mutations, right? Which is this paper right here. From this analysis, what they saw was how a specific mutation called A82V potentially is correlated with higher infection and higher deadliness of this strain that does have this mutation. So here, let's briefly review this. The magnitude of the 2013-2016 Ebola virus disease epidemic enabled an unprecedented number of viral mutations to occur over successive human-to-human transmission events, increasing the probability that adaptation to the human host occurred during the outbreak. We investigated one non-synonymous mutation, Ebola virus glycoprotein GP mutant A82V, for its effect on viral infectivity. This mutation, located at the NPC1 binding site on Ebola virus glycoprotein, occurred early in the 2013-2016 outbreak and rose to high frequency. We found that GPA82V had heightened ability to infect primate cells, including human dendritic cells. The increased infectivity was restricted to cells that have primate-specific NPC1 sequences at the Ebola virus interface, suggesting that this mutation was indeed an adaptation to the human host. GPA82V was associated with increased mortality, consistent with the hypothesis that the heightened intrinsic infectivity of GPA82V contributed to disease severity during the Ebola virus disease and epidemic. Okay, so here we have an example of a specific mutation that is related to the origin of where the virus emerged from. And we have a sense of viruses being present in the reservoir host, which are bats, but then a spillover event that goes from bats to primates and eventually to humans, right? To first we have non-human primates, monkeys, and then it goes into humans. And this specific mutation might explain how this intermediate host plays a major role in how the virus behaves when it enters the human population. So by jumping over first into non-human primates, it actually became very effective at being transmitted between inside the human population. Now, this specific mutation continues. There's more and more discussion about this. And so related to this topic, I wanted to ask you to do the following. Okay, so we'll talk about, so again, we talked about recombination, we talked about diversity of viruses inside a host, and we talked about very specific mutations. All of these three aspects are important to understand how the virus goes from a host to an emerging disease to an epidemic or eventually to a pandemic. But to study this more closely, I wanted to ask you to do the following. Read the publication, Ebola virus glycoprotein with increased infectivity dominated the 2013-2016 epidemic. Okay, so this paper that we just looked at, if you can read this paper, that will help you understand what exactly they did and see how the methods that we have learned about could be applied to a real situation. And then if you could answer these 4 questions. Number 1, how many samples were analyzed? Number 2, what 2 major lineages were found? Number 3, which types of mutations helped characterize the 2 lineages? And 4, what might be the role of a specific mutation in the surface glycoprotein described in this paper? So actually, what is the function? Not just what mutation it is, what might this mutation mean for how the virus infects the human cells. Okay, so let's put this on the forum. Find the forum here so that all of you can think about this for the next time that we meet. Okay, does anyone have any questions about these topics? Yeah, go ahead. Go ahead, Roland.
You hear me, sir?
Yeah, now I can.
Yeah, you talked about like the selective mutations, like random and selective, and the evolution of Ebola virus. For us humans, like can selective mutations be an advantage, like natural selection, in a way to combat the evolution of the Ebola virus?
Yeah, so a great question. So again, the question is whether specific types of mutations could be beneficiary or they could be less, right, causing the virus to be actually less infectious or less deadly, as opposed to what we see with some of these examples of mutations that make it more severe, right? That's the question?
Correct. Like like basically like can mutations provide like in terms of natural selection, in a way, can mutations provide an advantage to like a disease like Ebola?
Yeah, so, definitely when we talk about how the virus mutated over time, we are talking about mutations that both made it more deadly in the very beginning, and perhaps towards the end, it actually reduced the virulence and made it maybe not as deadly, right? So the mutations can go both ways. Evolutionary speaking, for the virus, those mutations could be important because the more the virus can infect the host without killing the host, potentially makes the virus more persistent and more able to reproduce without causing death, which actually might make it much more difficult to contain, right? If you think about flu, flu does not typically kill the host, but it continues to reappear all the time. And so this ability to circulate within the population without killing the population, evolutionary speaking, might be more beneficial to the virus in some cases. So I think that mutations, when we look at them, there could be mutations that are beneficial to virus replication and cause death of host, because maybe it causes cytokine storms, maybe it causes severe tissue damage, maybe it causes severe organ collapse and things like that. But in some cases, some of those are actually going to be beneficial because they make the infection itself milder. Maybe the replication rates are not as high, but they nevertheless continue to spread between different humans, let's say, and therefore the virus becomes a persistent infection. And that persistent infection at a milder rate can continue for a much longer time throughout the lifetime of a human. For example, if you think about hepatitis or HIV, those infections last a much, much longer time, and they're spread in many, many more numbers without necessarily causing immediate death. Okay, and then Trinika was also asking, will the recording be available? And yes, definitely, the recording will be available, just like every time. And also, if you're able to look on the forum, I am posting some of these questions, and these questions can help you think about some of these topics that we talked about today. Yes, Deidre also wanted to ask something.
Yes, sir. When we were speaking in the assignment, when you're asking about the major lineages, did you want a title or a name in particular?
Yeah, that's a good question. So actually, if you look at the paper again, in this paper, you'll see that they actually specifically mention these lineages. and you know, the lineages eventually over time will be named. So actually, when we talk, I wanted to briefly talk about the coronaviruses, a similar analysis of coronaviruses, which would have addressed one of the questions that we had in the beginning about a new lineage. I'll just reference that briefly, but what you'll see in these studies is that eventually, as enough data is being compiled, the lineages become named. So I'll just focus on this slide. So this slide shows how we have specific lineages, right? So back in, I would say this was, yeah, so April. In April, for the novel coronavirus, you had 3 different lineages that were established: a V, an S, and a G. Before that, probably around February, there were only 2, the L lineage and the S lineage. You can see right here, S clade remained, but then the other ones actually became more diverse. And later on, so this is recent, this is from June, you can see that now we have the S, the L, V, and then the G split up into multiple, GH, G, and GR. So you now have 7 or 8 different lineages. So typically these lineages are associated with specific mutations that are consistent within this group of viral genomes, and they do get a name. And so those names is what I'm looking for. But you know, if you just say, okay, it was 3 lineages, and you know, one was characterized by this mutation, other one was characterized by another mutation, right? So either one is fine. These are actually questions that could guide your, you know, kind of thought about the problem and about the data that we have to answer the question. They're not necessarily assignments that, you know, you're required to complete and submit in some way. This is more for facilitating a discussion in our next session. Okay.
I think those are the mutations are occurring quite fast. You know, I think it's very fast. Typically, to me, I would think that it would take a longer time before the mutations started to appear. However, it just seems like it's fairly quick.
Well, right. These are RNA viruses, and both Ebola virus and the novel coronavirus are both RNA viruses. And RNA viruses are known for their high rate of mutation, right? So compared to, let's say, humans, those mutations are much more rapid. And we can see that the strategy of these viruses is actually by accumulating mutations and also recombination. And those 2 aspects of mutations kind of appearing in specific regions to help the virus evade the immune system, to help it adapt to new tissues and infect new tissues. Here in this example, you can see how those lineages originally separated between the S and the L lineage, and then the S practically, you know, was kind of minimal towards today. And then all the other ones gained more prominence, right? So today you can see that the G is the more prominent lineage compared to all the other ones that we had: V, B, and A, and O stands for other, right? So the proportion of other is also fairly small. So yes, the rate of mutation is very fast for viruses, and the establishment of these specific strains over time, you can see how it changes with the amount of data that is being generated, and also as the virus spreads to very different locations and very different populations. So specifically about the question that was posed in the beginning, I'm just going to finish with this. There was a recent publication that highlighted a specific mutation. The mutation is D614G, which means that the amino acid D changed to G in the position 614. in the SARS-CoV-2 spike protein. So the spike protein is a protein that we're going to turn to next. We talked about the Ebola glycoprotein. The Ebola glycoprotein is the surface protein on the Ebola virus that helps us and that helps the virus enter the human cell. And the coronavirus also has a glycoprotein, also responsible for cell entry. So you can also look at how specific mutations in this spike glycoprotein actually helps it become more dominant over time and potentially enhances viral transmission. So this is a publication. I'll post a link here. It's very new. It's still not peer reviewed, but I think an interesting perspective on how the same dynamic that we saw in the Ebola virus actually is happening in the coronavirus pandemic. Okay, so I don't want to take too much more of your time. I know that we're again over the hour that is allocated for the session. I did have a whole section here on coronaviruses that I wanted to start talking about and actually have a brief assignment, but I'll continue with that next time. If you want to kind of get ready, I highly recommend going through the video that I mentioned in the beginning. That video on Friday, we had a session with Dr. Kasoulis, and let me, I believe the video recording is also going to be available on the forum. Let's see. Dr. Kasoulis spoke about the specific biology of coronaviruses, and also about the different strategies for vaccine design and antiviral drug design. And I also talked a little bit about the evolution and the, okay, so it wasn't posted here, but I'll post it. It's actually in the activity tab. I mentioned some of the things related to how the virus evolved once it entered into humans, and also some of the potential origins, which we'll talk about next time. So let me briefly outline, and this is the video. So please take a look at this video where we talk about those things. And briefly, let me show you some of the things that we'll try and talk about in our next session, quickly introducing the problem with coronaviruses and how recombination in the host and the similarity with the host might be a factor to consider. So we have this video. In this video, we see different hosts and the different intermediate hosts for the different coronaviruses. We'll talk about how we can identify a potential intermediate host and how we can study the closeness of relationship between the virus that we observe in the human population and the intermediate host. We'll talk about it in the context of multiple sequence alignment that allows us to study evolutionary divergence, how different viruses are different from in different hosts or between different outbreaks. And we already alluded to the different outbreaks when we did this analysis and comparison between beta coronaviruses that, remember, don't really cause severe symptoms. MERS, that had an intermediate host of camel, but also originated from bats. SARS-1, that we do know an intermediate host of, and the intermediate host of SARS-1 was the civet cat, which is really like a feline, but specifically in Hong Kong. And COVID-19, for which we actually do not know the intermediate host. So by doing this analysis, we can identify differences between the viruses in terms of where they emerged from, and then how did they evolve over time in the human population. And this is more of a view of how they evolved in the human population and specific mutations that might play a role in the way the virus infects the human cells. By looking at the comparison between SARS and SARS-2, we can identify specific patterns. And we can also look at the divergence between different hosts. So we actually have viral sequences like we saw in bats. We have viral sequences in pangolins, which might be an intermediate host. And we can know specific viral sequences in the early samples taken from human patients. So we'll review some of this and we'll look at how to find data about the coronaviruses that can help us answer some of these questions. Look at similarity. Here you can see bat coronaviruses and other coronaviruses, either more closely related or farther away from the human coronaviruses, COVID-19 causing coronaviruses. And we'll look at how these different lineages are related to similarity, the similarity in specific regions of the genome. And those regions are actually coding for this spike the spike glycoprotein from the coronavirus. And we'll see how bat sequences, pangolin sequences, and human sequences might point to the specific mechanism of action for this coronavirus protein, which you see right here. And we'll try to answer some of those questions together in the next session. And then I'll have an assignment for you to think about the role of this protein that we will continue to explore in the next session, because this specific protein is the reason for the virus to be able to enter the human host. And one of the biggest questions that we have is, how did it evolve to acquire mutations that allow it to enter a human cell when the host receptor is so different between the human and the pangolin or even the bat receptors. And we'll look at the structural alignment of those different proteins and understand how did that exactly happen. Okay, so if you have any other questions, please make sure to post them on the forum. I will add the recording of today's session and the Friday webinar recording so that all of you can take a look if you're interested, and also post links to some of the papers that we discussed today, the paper about Ebola viruses, and the paper that it would be beneficial for you to look through for the next session. Okay, are there any questions? Okay, and again, if you could take a look at your own individual progress, just to kind of point out, we are anticipating that some of you will be more interested and will have more time to work on an independent project. If you are interested in working on an independent project, I highly recommend going through some of the introductory materials that are posted in the courses and the projects. And that will prepare you for, selecting the data sets and doing the analysis independently. And we'll go through several sessions dedicated to project design and proposal so that you can prepare that project. So I'm looking forward to seeing you again next time. And in the meantime, if you have any questions, like I mentioned, please post them on the forum so I can prepare the answers between now and the next session that we'll have. And the next session is going to be on Thursday, I believe. So if you go to the homepage, we do have our next session on June 25th to 22nd. Okay, so great to see you again, and I'll see you again next time.